Hello viewers, welcome to my YouTube channel. Today our discussion topic is structure of a bacterial cell. Bacteria comes under the group Monera. The Monera group that is characterized by prokaryotic unicellular. So bacteria is a prokaryotic cell and it's a unicellular. So today we are going to study about the structure of a prokaryotic cell. Okay. So here is the bacterial cell. A cell has its outer covering and inner structure, outer covering cover structure and inner intercellular cell organelles. So outer covering has outermost covering. There is a layer. This layer I separate that part by yellow color. This part is called the glycocalyx. It's a layer surrounded by this layer surrounds the bacterial cell. This layer is called glycocalyx. Glycocalyx is actually made up of polysaccharides, protein. So even if it's a slimy, if it's a soft layer, then it's called a slime layer. And if that covering is tough, then it's called capsule. It protects the bacterial cell. Glycocalyx, the main function of glycocalyx is to give the protection to the bacterial cell. So it is the outermost covering of the bacterial cell. If it is soft, then we call them as a slime layer, and if it is hard, then we call capsule. Next to the glycocalyx, there is a cell wall. This brown part is the cell wall. Bacterial cell wall is made up of peptidoglycan. Next to the cell wall, there is a cell membrane. This is cell membrane. Cell membrane or also called plasma membrane. Plasma membrane. Cell membrane or plasma membrane. Okay. Now there are some other structure at the outer covering, that is from this part. This is called the flagella. Okay, that arise from the plasma membrane, that part. A flagella has three parts. This is the basal body. This part is called hook, and this is the filament. So flagella is the locomotory organ of the bacteria. Bacteria possesses flagella for their locomotion. They possess flagella, cilia, fimbriae and pili. Pili is very small in structure and numerous in number. The small part, small locomotory organ. Actually, flagella is the locomotory organ, but pili, cilia and flagella. This is cilia. This is flagella. These three part comes under filament, hook and basal body. This is flagella. And this is cilia. Cilia and flagella, these two are locomotory organ. And there are another two structures. This is the pili and pili, which is made up of pillin protein. Pillin protein. And this is fimbriae. Fimbriae. So this pili and fimbriae helps the bacteria to adhere to the substratum. Whenever bacteria come to adhere on the substratum, they use fimbriae and pili. So these two are the structural element that helps the bacteria to adhere to the substratum. But cilia and flagella, these two are the locomotory organ. They help the bacteria to move from one place to another. So these are the four structures we can find at the outer covering. Flagella, it has three parts: filament, hook and basal body. Okay, so now we are coming to the inner part of the bacteria. Look, here is the plasma membrane. That plasma membrane fold at some points and form a structure called mesosome. Mesosome. Okay. Now I want to focus on the cell wall of the bacteria, because depends on the cell wall, then I will come on the inner part. Based on the cell wall, bacteria are of two types, that is gram positive bacteria and gram negative bacteria. Okay, now come in detail. Here is the structure of the cell wall of gram positive bacteria, and this is gram negative bacteria. In both of the cases, this is the plasma membrane. At the lowermost part, this is plasma membrane. Okay, and above the plasma membrane there is a space. Here the space is very little. Okay, here is the space. This space is called periplasmic space. Periplasmic space. Periplasmic space. And then there is a peptidoglycan layer. Look at one thing. For the gram negative bacteria, the layer of the peptidoglycan is thin. But in case of the in gram positive bacteria, peptidoglycan layers are thick. Gram negative bacteria has thin peptidoglycan layer. Gram positive bacteria have thick peptidoglycan layer. Okay, another difference is that for the gram negative bacteria, they have another membrane. That membrane is called the outer membrane. This is outer membrane. Outer membrane. That can only found for the gram negative bacteria, but gram positive bacteria don't have any outer membrane. Okay. And above the outer membrane, they have phosphoglyceric, phospholipids, glycolipids. These are phospholipids and glycolipids. And these are some internal integral proteins as present in within the plasma membrane. So this is the inner plasma membrane, outer membrane, inner membrane or plasma membrane. Then there is a periplasmic space, a space between the cell wall and the cell membrane. There is a space, that is periplasmic space, and the liquid present within it called the periplasmosome. Periplasm. Okay, the liquid present within the periplasmic space is the periplasm. So now look at the structure of the peptidoglycan layer. Peptidoglycan layer is a chain of the oligosaccharide chain made up of proteins and polysaccharides. There are N-acetyl glucosamine, N-acetyl uramic acids. These are alternatively arranged and form a chain, and that chains are linked by a cross bridge protein. That's why the cell wall of the bacteria is very rigid and give protection to them. Along with the thick peptidoglycan layer, gram-positive bacteria also possesses a structure. That structure is called the teichoic acids. Teichoic acid. Okay, so now revise once again. Cell wall of gram-positive and gram-negative bacteria. Gram-positive bacteria have thick peptidoglycan layer and very thin periplasmic space. They have gram-negative bacteria, they have thin peptidoglycan layer and more periplasmic space. Along with these structures, gram-negative bacteria have outer membrane. Outer membrane is linked with phospholipids, glycolipids, and in case of positive bacteria, they have a special structure called teichoic acids. Okay. This name gram-negative and gram-positive comes by the name of the scientist Christian Gram. When we actually stain bacteria by crystal violet, and there are several steps for staining, but when we stain by bacteria cell by crystal violet, and there is a step when we have to wash bacteria, we have to wash the stain by alcohol. So, for the gram-positive bacteria, if we wash the stain by alcohol, the crystal violet stain retained within the cell, and under the microscope that is look like dark violet in color. But in case of gram-negative bacteria, if we wash the gram stain, crystal violet stain by the alcohol, all the stain washed out from this space. As because the peptidoglycan layer is thin, so all the stain that entered within this space that washed out by the alcohol. So the color is not dark when we see the bacteria under the microscope. Depends on this procedure, the Christian Gram classify these bacteria into two groups, that is gram-positive bacteria. They retain the gram stain as because their peptidoglycan layer is very thick. The stain not able to get out of the cell wall. But here gram-negative bacteria, as they have thin thick peptidoglycan layer, they don't have they are unable to retain the stain. All the stains get washed out by the alcohol. Okay, so these are the two types of bacteria. Now come to the inner part, the inner cytoplasm. Here is the nuclear material called the nucleoid. Okay, this is nucleoid, and the cytoplasm inside them is called the xenophore. Cytoplasm inside them is called xenophore. Now, this is the ribosome, and it's a 70S ribosome. We can find in case of prokaryotic cell. And they have a chromosome. Nuclear material is actually a chromosome, and along with this nuclear material, they have another circular DNA. That circular chromosome, that circular DNA is called the plasmid. This is plasmid. Okay. One thing we can find in prokaryotic cell, that is on the mRNA, more than one ribosomes can attach and form a polysome, polyribosome or polysome. That structure we can find in the prokaryotic cell. There are another structure we can find in the bacterial cell, that is we call them as a chromatophore. That cell can store the chlorophyll inside it. We can find in this structure in the photosynthetic bacteria. So this is chromatophore. Chromatophore. Okay, so these are the thing we can find, and another thing is that this is the stored food. Stored food. It stores glycogen, synophysin, starch, phosphate, granules. So these are something stored within the bacteria. This also can find in the blue-green algae. Okay, so these are all about the bacteria. So now what we learn? Bacteria is a monera. Under the group monera is a prokaryotic unicellular cell. At the outermost covering there is a layer, that is glycocalyx, that give protection to the bacteria. Below the glycocalyx there is a cell wall, a thick cell wall made up of peptidoglycan, and depends on the thickness of the peptidoglycan. The bacterial cell is of two types, gram positive and gram negative. Then at that portion there are two locomotory organs, cilia and flagella. Flagella made up of flagellin protein, and this is cilia. Flagella is of three parts. Flagella has three parts: basal body, hook and filament. And another two structures are there, that is fimbriae and pili. Pili is the smaller one. Fimbriae is longer than the pili. Pili made up of pilin protein, and this is fimbriae. They are mainly responsible for adhering the substratum. They support the bacteria to adhere. Pili is are of two types. Vegetative pili is actually help the bacteria to adhere to the substratum, and there are another pili also. They help the bacterial cell. Two bacterial cell. Suppose this is two bacterial cell. When at the time of reproduction, these two bacterial cell come together and joined by their pilis. This pili is called the fertility pili or sex pili. Fertility pili or sex pili that help in the formation of conjugation tube. By that tube, one bacteria can push their genetic material to the another cell. So this pili is help them to form the conjugation tube. So pili is of two types, vegetative pili and sexual pili. Vegetative pili help them to adhere to the substratum, and reproductive pili, sex pili or fertility pili help them to form the conjugation tube. And there is a cytoplasm inside the cell. Within the cytoplasm there are some cell organelles. As it's a prokaryotic cell, so they don't have a definite nucleus. A primitive nucleus we can find here, and this is the nucleoid, the primitive type of nucleus, not well defined nucleus. The nuclear material inside it is the xenophore. And also there is a mRNA. On the mRNA there are more than one ribosomes, that they are called polysome. This form is called the polysome, and the ribosome is of 70S. The smaller subunit is 30S, and the bigger subunit, larger subunit is 50S. And there is a structure that store chlorophyll for the photosynthetic bacteria. This structure is called chromatophore. And in some places the inner membrane of the bacteria get fold inside, and they form the mesosome, that actually responsible for storing some respiratory enzyme. Mesosomes are responsible for storage of respiratory enzymes. So these are all about the bacterial cell. Thank you for watching. We will meet soon with new topic on my next video. Till then, take care.